This lecture video will cover Chapter 24, the urinary system material, specifically physiology of urine formation and filtration. So the outline for urine formation, there's going to be 3 different steps that we'll discuss. So it starts with glomerular filtration, which is highlighted. This is all that will be covered in this video. And then tubular secretion and reabsorption will be in the next video. So like I just said, there's going to be 3 different processes that we go through in order to actually form urine. So the first process is all we're talking about in this video is glomerular filtration. This is occurring here in that renal corpuscle. So we're going to be filtering substances out of those glomerular capillaries. It's going to get contained in this capsular space, and that fluid is then going to be called filtrate. From there, we're then going to go on to these next 2 processes here, tubular reabsorption and secretion. These do occur simultaneously. We just put them in an order so that we'd have a nice process to work our way through in this lecture. So both reabsorption and secretion occur in the renal tubule segment, so PCT, DCT, and nephron loop. So I really want you guys to understand the difference between reabsorption and secretion. We're going to talk about this a lot for the rest of this chapter. Reabsorption, we see here in this diagram is with blue, anything that is going back into the blood. So we are reabsorbing it, we are keeping it. That's how I remember reabsorption. So we're going to reabsorb a lot of water, a lot of salt, glucose, stuff like that. Secretion is going to go from the blood back into the tubules, because remember, at the end of all of this in that collecting duct, that's where we get urine, and urine is excreted from our body. So excreted, secretion, those kind of sound the same. Anything that stays in those tubule segments and doesn't go back into the blood, that's getting eliminated. So reabsorption goes back into the blood, secretion goes from the blood into our tubule segments. We're going to spend a lot of time on that. We're hopefully going to be very familiar with those words by the end of this chapter. I really like this figure here because if you haven't noticed so far, I'm a huge fan of color coding things, and this image color codes very nicely. So we are looking at our nephron here. We see that renal corpuscle, proximal convoluted tubule, loop of Henle, distal convoluted tubule, collecting duct. So I like how they outlined all these processes of urine formation. So number 1, glomerular filtration, what we're talking about today, is a green arrow. So I see that green arrow going from our glomerulus into that capsular space. Then I see number 2 and 3. So 2 is reabsorption, and that is in purple arrows. We see this all throughout the tubule segments. It is going from the tubule into the blood for all of these cases, no matter where it is. Then for tubular secretion, this is in yellow. I see this going from the blood into the tubular segment in all cases. So this is very important for us to remember. As we go throughout what we're covering in this lecture video, glomerular filtration, this is only occurring in the renal corpuscle. This does not happen in any of the tubular segments. So let's go ahead and talk about filtration. All right, so what is filtration? I told you guys all of these pressures were going to come back to haunt you. Well, here they are. So glomerular filtration should hopefully actually be a pretty passive process, one that happens very easily without much energy. And this is due to a hydrostatic pressure. We're going to have a very high hydrostatic pressure in our glomerular capillaries that's going to push fluid out of those capillaries into the capsular space. We are going to have, call it osmotic pressure. We'll have hydrostatic pressures of different areas, so be prepared for that. But this fluid that we are getting from those glomerular capillaries and that's going into the capsular space right here, that is called filtrate. It is not blood, it is not blood plasma, it is not urine. It is called filtrate. And we're going to talk about what filtrate is soon. So how are we actually creating this filtrate and having filtration occur? We have something very special in our renal corpuscle called the filtration membrane. This is going to be between the glomerular capillaries and the interior of the glomerular capsule. So So looking at this image over here, it's showing us the filtration membrane. What we see over here is everything that's contained in the blood of a glomerular capillary. And this right here says the endothelium of our glomerular capillary. Then, do you guys remember the cell types that make up the visceral layer of the glomerular capsule? Hopefully you said podocytes. So all these cells right here, these are actually those podocytes. So that's going to be that visceral layer of our glomerular capsule. So this is where that filtration membrane is, and it's only going to allow select things to pass from the blood into that capsular space. So this filtration membrane, like we see everything bold here, it's going to be porous to allow things to enter. It's going to be thin, just like we saw when we had gas exchange at the respiratory membrane. If we're exchanging things back and forth across the membrane, we typically want it to be thinner. It's easier for things to pass through. And it's also going to be negatively charged. We'll talk about why that's fancy pretty soon here. So this is going to allow us to actually filter water through this space and anything that is not a very large plasma protein or a blood-formed element. So no white blood cells, red blood cells, platelets, or large proteins are going to get filtered through this membrane into the capsular space. There's going to be 3 different layers that help compose this filtration membrane. We're going to have the endothelium of the glomerulus or glomerular capillaries, the basement membrane of the glomerulus or glomerular capillaries, and that visceral layer of our glomerular capsule, those podocytes. So this image right here, hopefully you guys all have this out. This is from your chapter 24 physiology diagrams. This is the filtration membrane picture. So before we get into all this, let's kind of label what the heck is going on here. So this whole space right in here, don't color this in on yours, you'll just mess it up. But this whole space is a glomerular capillary. So I'm going to erase that real quick. And this here is a glomerular capillary. So all of those glomerular capillaries make up the glomerulus. We see here, these are those endothelial cells of the glomerular capillaries, and they are fenestrated. So that's going to allow things to pass through them. Feeding into the glomerulus, remember, is the afferent arteriole, and draining the glomerulus is the efferent arteriole. I need to fix that on your drawings. Should be efferent arteriole. Then looking at this weird blue thing over here, what this whole thing is supposed to represent is this is going to be representing the parietal layer of the glomerular capsule. And then we see here, I'll just go ahead and fill this in right now. What this is pointing to, number 3 specifically, is those podocytes, but this is that visceral layer of the glomerular capsule. So remember, this is composed of podocytes specifically. So that's what these cute little black guys are supposed to be there. So that space that we form in between the parietal and visceral layer of the glomerular capsule is right here, that capsular space. So everything that we're going to contain in this space is going to be called filtrates. So we're going to be filtering stuff from these glomerular capillaries into the capsular space. Let's go ahead and label the other 2 things that make up our filtration membrane. So arrow number 1 here, these are the endothelial cells of our glomerular capillaries. So endothelium of the glomerulus, and you can put glomerular capillaries, really whatever you want. We will have different areas of the filtration membrane for each of those glomerular capillaries. Then number 2 right here is going to be the basement membrane of the glomerulus. So that's what that guy is there. All right, now I want to do this little exercise with you guys. I know that we're not actually meeting, but hopefully you can answer along with me as I go through this. So everything that's inside this glomerular capillary is considered blood at this point. We're going to have a lot of things in there. So does anybody remember the 3 blood-formed elements? I'm going to write B, F, E for blood-formed elements for the sake of space. I'll draw one of them out right here, maybe. Alright, that's supposed to be a red blood cell. This guy right here should be white on the inside. Alright, this is a white blood cell. And then we also have those cell fragments. Does anyone remember what those are? Hopefully you all said platelets. Hopefully you guys are all talking out loud and anybody else that's with you in quarantine is laughing, but maybe learning something. Alright. Other things that we're going to have in our blood are going to be large plasma proteins. So plasma proteins are things like immunoglobulins or our antibodies. Now, does anybody remember what the most common immunoglobulin or antibody is in the blood? Hopefully at least someone said IgG. So I'm going to write large plasma proteins here in purple. So we're going to have IgG. And then, does anyone remember the most common plasma protein? It was an exam question. It starts with an A. Maybe someone said albumin. So we are going to have larger plasma proteins. Obviously, there's more than just 2, but those are nice examples and kind of a review to tie everything we've learned together. Inside our blood, we're also going to be carrying a lot of nutrients. I know we haven't talked about digestion yet, but hopefully somebody can think of some nutrients for us. So really big one, our body's main energy source. Does anyone have an idea? Hopefully someone said glucose. So we're going to have glucose. We're going to have things like lipids or fats, amino acids. These guys are the building blocks of proteins. Another nutrient that we can talk about is actually water. Water is considered a nutrient. We're also going to have a lot of vitamins and stuff in here. Another thing that's going to be really important that's floating around in our blood is going to be different ions. Does anyone remember the ion that's responsible for depolarization and action potentials? Hopefully someone said sodium. So that's Na+. What about the ion that's responsible for repolarization and action potentials? Hopefully someone said potassium. That is K+. And then I'll just write these out for you guys now, things like calcium, chloride, magnesium. I know we haven't talked about bicarbonate yet, which is HCO3-. This is going to be really important in acid-base balance, which is our next chapter. So the reason why I have color-coded all these things the way I have is because everything that is this orange-yellow color, that's the same color as the filtrate, this is going to be the stuff that is allowed to pass through this filtration membrane. So all of these ions can make their way through. All of these nutrients can make their way through. However, these large plasma proteins and our blood-formed elements cannot pass through that filtration membrane, and they are going to continue on in the efferent arteriole, and they don't get filtered through into the filtrate. So that is why this is called filtrate and not blood. It's not blood plasma. It's very similar in content, but it is not. It is filtrate. From here, this then goes into the proximal convoluted tubule, where that fluid changes name again. Does anybody remember? We covered it in the last video. That is then going to be called tubular fluid once this fluid enters into the tubular segments. So we have filtrate in the capsular space of our glomerular capsule. Now we are going to talk about the different layers of our filtration membrane specifically. So looking at these, they are numbered from innermost to outermost. So innermost is closest to the glomerulus or the glomerular capillaries. So this first one is going to be the endothelium of the glomerulus. Like I said earlier, this is going to be fenestrated. It's going to have those little pores between the endothelial cells, and this is going to allow blood plasma, so that water component of blood, and dissolved substances, so things like those ions and our nutrients can actually pass through these fenestrations. But really large structures like erythrocytes, our red blood cells, and our white blood cells are not going to be able to make their way through these fenestrations. What can make its way through these fenestrations are going to be larger plasma proteins, so maybe things like IgG can get through. So that's going to lead us to this next portion of our filtration membrane, which is the basement membrane of the glomerulus. So this is going to be in between the glomerular endothelial cells and the visceral layer of our glomerular capsule. So looking at this image over here, it's this portion, this kind of lighter peach portion. And what this is allowing us to do is it actually forms a physical barrier that is going to block any of those large plasma proteins. Also, another really cool thing about this layer is that it actually has a negative charge. So if you guys know anything, I guess, about charges, you probably heard that saying, opposites attract. So if we had a positive charge and a negative charge, those would be drawn to each other. Most of our proteins are going to have a negative charge to them, and then that basement membrane of the glomerulus will also have a negative charge. This is going to cause them to repel. So this is actually going to physically repel a lot of those proteins and therefore blocking them from being able to get filtered into that glomerular capsule. Let's go ahead and draw in these specifics on our diagram here. So first, let's talk about the endothelium of our glomerulus. What I want you guys to know about this is that it is fenestrated. So this is going to allow us to block those blood-formed elements. Then our next layer right here, that basement membrane of the glomerulus, this little kind of dotted line right here, I know that looks kind of weird, that is indicating the negative charge of that basement membrane. So remember, most of these larger proteins are going to be negatively charged as well. This is going to repel those large proteins. So that negative charge is going to repel those large plasma proteins. So those aren't going to be able to pass through. Now let's go ahead and talk about that last layer. So that last layer is going to be the visceral layer of the glomerular capsule. So remember that visceral layer is composed of those podocyte cells. Oh, I thought I had black, but blue is fine. So those podocyte cells, remember, they're kind of those fancy foot-like cells. And if we look at this image down here, we see that this is a nucleus. So all of this is one large podocyte. This is going to be the glomerular capillaries. We see those fenestrations, those little holes. Then looking at this podocyte, we see filtration slits. So those little spaces in between where we kind of see the red of that glomerular capillary peeking through, those are those filtration slits. And that's really going to restrict the passage of any of those smaller proteins that might have made their way through here, because in our filtrate, we do not want blood-formed elements and we do not want proteins. So if any of those smaller proteins were able to kind of sneak out of those fenestrations of the glomerular capillary endothelial cells, they weren't as necessarily repelled by that basement membrane being negatively charged. And now this visceral layer of the glomerular capsule, these filtration slits are going to be far too small for those small proteins to even squeeze their way through. Within this visceral layer of our glomerular capsule, we also have something called mesangial cells. These feel very similar to macrophages. They are phagocytic. Something that's really cool is they are contractile, so they can actually make these filtration slits even smaller, and they can release some signaling molecules kind of to tell the rest of those podocytes to kind of have contraction and decrease the size of those filtration slits. So it's actually a really cool physiologic process, but you guys don't need to know the details of that. What you really need to know about this visceral layer of the glomerular capsule is that it's composed of podocytes that have those filtration slits that will block the passage of most of our small proteins. So now we can finish this figure for us. So going up to number 3, this visceral layer of our glomerular capsule. So remember, this is made up of podocytes. And these podocyte filtration slits, so filtration slits, are going to block small proteins. So we have blocked those smaller proteins at this point. So just kind of a nice review of all these layers of our filtration membrane. over here, number 1, we have the endothelium of our glomerulus, of those glomerular capillaries. It is fenestrated. It has those little pores in between it that are going to block blood-formed elements from being able to pass through here. Next, we are going to have number 2, this basement membrane of our glomerulus. It has a negative charge. This negative charge is going to repel any of those larger proteins. Then that third layer is the visceral layer of the glomerular capsule. It's composed of those podocyte cells that have those little filtration slits in between them, and this is going to block any small proteins from entering into the capsular space. So all this stuff again over here that's in orange, this is the only stuff that's going to be able to get through all 3 of those layers. So those nutrients and those ions, remember water is considered a nutrient, is the only thing that we want to have in our filtrate. So filtrate is the name of the fluid in the capsular space. So I'm going to go ahead and write this in black so you can really see it, because yellow on yellow is hard to see. So that filtrate is going to contain no blood formed elements and no proteins. We do not want that in our filtrate. And then again, we've already written this in, but after that fluid flows into the proximal convoluted tubule, it is then called tubular fluid, not filtrate. So again, kind of going over what filtrate is. It is derived from blood plasma, but it is not blood. You should not have any blood in your urine because that filtrate, we want no blood-formed elements. We do not want red blood cells, white blood cells, platelets. If that is showing up in your urine, that is indicative of probably some sort of glomerular filtration problem. We also want no proteins in there. That is bad too. So star this. I want you to know that our filtrate contains no protein and no foreign blood elements. Definitely need to know that for exams. This number up here, we do produce about 125 liters of filtrate a day. That's a ton. So remember, that is why I spent so much time going over the blood supply to our kidneys, because this is blood plasma derived fluid. So remember, it's called filtrate when it's within the capsular space. Then it gets funneled to the proximal convoluted tubule, where it is then tubular fluid. So any of the materials left in the blood that don't actually get filtered into the capsular space as filtrate, those are going to continue on in the efferent arteriole to those peritubular capillaries. So remember that. And these guys right here, those mesangial cells, are phagocytic. So if there's anything kind of trapped in that basement membrane that we don't want, those cells will phagocytose it and get rid of it. All right, this is where all of those different pressures are going to come into play. So this is a very important job of our kidneys. This is the first step of urine formation. So you can actually diagnose a lot of disorders and make sure someone is healthy by being able to calculate their glomerular filtration rate, or GFR. So by definition, it's going to be the volume of filtrates that we form each minute by the combined activity of all 2 million of our glomeruli. So remember, each kidney is going to have about 1 million. So that's why we have 2 million, because we're talking about both of our kidneys here. So a normal glomerular filtration rate in a healthy individual is going to be about 125 milliliters a minute. So this can change if we have really low blood pressure, really high blood pressure, if a certain area of that filtration membrane is kind of broken down or not working. So this is a really good way for us to kind of be able to understand what's going on in the body. This is going to be directly influenced by net filtration pressure. So this is the pressures, or NFP. So if we have an increased net filtration pressure, that's going to increase that glomerular filtration rate. It's going to push more fluid from those glomerular capillaries into the capsular space. So this whole equation right here is going to be the equation for net filtration pressure, or NFP. So this is where hydrostatic pressures come into play, our colloidosmotic pressures come into play. This time I am really going to make you understand what these pressures are doing. However, again, this is not a math class. I'm not going to make you memorize an equation. I'm not going to make you use a calculator, but you have to understand how these variables relate to each other and what they mean and how they affect net filtration pressure. So let's first talk about kind of just overall, we can classify these as outward and inward pressures. So outward pressures for us are going to promote filtration. They are going to push fluid out of the glomerulus. These inward pressures are going to inhibit filtration. They are going to push or pull fluid back into the glomerulus. So this outward and inward pressure definition, these have come from talking about this in relationship to the glomerular capillaries or glomerulus. So if something is going out of the glomerulus, it is filtering out into the capsular space. If it's going back into the glomerulus, that is the opposite of filtration. It's getting basically reabsorbed back into the blood. We don't want that. So we are going to have two pressures that fall into each of these categories. For our outward pressures, a really big one that influences it is this first guy right here, this hydrostatic pressure of the glomerular capillaries. So hydrostatic pressure, just like when we talked about it before, this is abbreviated as HP. There's going to be a little underscore letter for all of these that's going to indicate what we're talking about. So when we talk about those glomerular capillaries, it gets that little subscript G for glomerular capillaries. I want to point this out because this other one here, this C, is for the capsular space. A lot of people get confused and they say the C is for capillaries, but it's not. So G is for glomerular capillaries or glomerulus. The C is the capsular space. So keep that in mind when you guys study. That's a very common thing I see people get confused. So that hydrostatic pressure of our glomerular capillaries is going to push fluid out of the glomerular capillaries into the capsular space. Then we have this colloid osmotic pressure of the capsular space. So colloid osmotic pressure, we are going to write as OP. You can also write this as COP if you want, but your text refers to it as just OP. So if you guys kind of remember from several lectures ago, this colloid osmotic pressure exists due to the presence of proteins. Proteins like to pull fluid towards them. However, hopefully we remember it was just a couple slides ago, what do we not want our filtrate to contain? We do not want it to contain proteins. So hopefully in the capsular space where we contain filtrate, there's essentially a zero colloid osmotic pressure in there because there shouldn't be any proteins, so that shouldn't even be an issue. So right here, hopefully it's zero in a healthy individual. You shouldn't have a colloid osmotic pressure of the capsular space. If this was here, this would cause us to pull fluid into the capsular space, but it's essentially zero. So now these inward pressures, these are going to inhibit filtration. So if we're inhibiting filtration, we're essentially making sure nothing is going into the capsular space. It's all staying in the glomerular capillaries. So we're going to have a hydrostatic pressure, HP, of the capsular space with that little c. So hydrostatic pressure is going to push fluid. So in this case, it's going to push fluid from the capsular space into the glomerulus. And we are also going to have a colloid osmotic pressure, or OP, of the glomerular capillaries with that little G. So what this is going to do is we still should have all those proteins, basically. All the proteins that we were talking about will still be in those glomerular capillaries. So those are going to pull fluid from the capsular space into the glomerular capillaries. Let's go ahead and fill out a little bit of this drawing here. So this, I think, is the last drawing that you guys have in your urinary physiology diagrams. It's called glomerular filtration. So let's go over a little bit what we're looking at here. So this bigger vessel is the afferent arteriole. Remember, that's going to be a vessel that's much larger in diameter. That feeds into our glomerulus, which is supposed to be this weird bubble-looking thing here. And then the efferent arteriole over here is where we are going to drain that blood from the glomerulus and those glomerular capillaries, and it is much smaller in diameter. This diameter is going to affect some of the things that we talk about today, so keep that in mind. This right here is our glomerular capsule. So I know it's kind of weird. You can't see the visceral and the parietal layer. I know that doesn't make sense. Let's just pretend we kind of cut away a cross section here, and what we're looking at in this area is going to be our capsular space. So in here is the capsular space, which is where we are going to contain our filtrate. So let's just pretend that's what that is. Then going on this way is the proximal convoluted tubule. So we talked about the outward and inward pressures on the previous slide. So our outward pressures are going to push fluid out of the glomerulus into the capsular space. So these guys are going to inhibit or promote filtration. They are going to promote filtration. So in this box you can write promote filtration. Then these inward pressures are going to pull fluid back into the glomerulus from the capsular space. So they'd be going in this direction right here, and these are going to inhibit filtration. So for now, let's just write out what these all are. So the two pressures that we talked about that are promoting filtration are going to be that hydrostatic pressure of the glomerular capillaries and the colloid osmotic pressure of the capsular space. So these other ones down here that inhibit filtration is going to be the hydrostatic pressure of the capsular space and the colloid osmotic pressure of the glomerular capillaries. So I'm going to go ahead and write in kind of what these would be about, what their millimeters of mercury would be. So our hydrostatic pressure of our glomerular capillaries is about 60 millimeters of mercury. You guys do not need to memorize that, but this is just for us to see this is a really, really high hydrostatic pressure. That's going to be important for us in a second. Then this colloid osmotic pressure of our capsular space. Remember, this is set up due to proteins. Do we have proteins in the capsular space? Hopefully not. So this is going to be about 0 millimeters of mercury. So I'm going to go ahead and fill this out already. If this did exist, this would pull fluid from the glomerulus. I'm just going to write glom for space. Glomerulus into the capsular space. However, there's virtually 0 proteins. I'm going to write capsular space as CP. CP. So this colloid osmotic pressure of the capsular space is not considered in our net filtration pressure, or NFP. All right, let's go to these last two down here. Our hydrostatic pressure of the capsular space is going to be about 18 millimeters of mercury. And our colloid osmotic pressure of the glomerulus is going to be about 32 millimeters of mercury. So now we are going to go ahead and we're going to fill out the rest of this as we go throughout the specifics of each of these pressures. So let's first talk about this glomerular hydrostatic pressure, or HPG. So this is the blood pressure that's inside the glomerulus. Like I said, this was about 60 millimeters of mercury. This is a really high pressure. The reason why this is so higher is that diameter difference between the afferent and efferent arteriole really does make a difference. The afferent arteriole is much bigger, so it's going to allow much more blood to be able to flow through. We're getting a lot of blood flowing through those glomerular capillaries, and then they have a very small place to exit. That efferent arteriole is very, very small. So I think I gave this analogy a little bit before. If anybody's ever been to a sports game or a concert or something, when everyone's trying to leave, you're going probably from a really large open area. A lot of you can flow into there, but then when you exit, there's usually a smaller door that everyone has to filter their way out of. That's essentially what's happening here. We're getting a lot of blood in an area, or a lot of people, and then that efferent arteriole is the really small exit door. So we're getting a huge buildup of people or blood in that larger area in the glomerular capillaries, and this significantly increases that hydrostatic pressure in our glomerulus. This is important because since we talked about that colloid osmotic pressure of the capsular space is the only other outward pressure to promote filtration, this pressure is basically the only pressure that we have that's going to promote filtration. Therefore, it's got to be super, super high, and if this is not high enough, we probably won't be having filtration. So by definition, our hydrostatic pressure is always going to push, and it's going to push fluid in whatever area we're talking about. So that's subscore or subscript G here. So it's going to push fluid out of the glomerulus into the capsular space. So let's go ahead and fill this out on our diagram. So I typed things up here just so it's clearer for you guys to actually be able to see what's going on. So let's talk about our hydrostatic pressure of the glomerulus. So like I said, it's about 60 millimeters of mercury, and this is going to push fluid out of the glomerulus into the capsular space. So let's go ahead and fill this out on our diagram over here. So this pressure exists in the glomerulus. So we're gonna want to have something in here. So I'm gonna write it in this box. So HPG. It is going to push fluid from the glomerulus into the capsular space. Also, this pressure over here, I realized that I didn't fill out the little box on our last slide, but this is our colloid osmotic pressure of the capsular space. So I drew the box in the capsular space because that is where that pressure would exist, and it is going to pull fluid from the glomerulus into the capsular space. But remember, this would be due to the presence of proteins. That's why I'm drawing all these dots, but we shouldn't have any of those in there. All right, let's continue on. So next we're going to talk about the hydrostatic pressure of the capsular space. This is going to be an inward pressure that inhibits filtration. So it is a hydrostatic pressure, still going to push fluid out of an area. So let's look at this subscript here. It's going to push fluid out of the C, the capsular space, into the glomerulus. All right, let's go ahead and draw this guy in now. So it's an inward pressure. It's going to be down here in blue. It's about 18 millimeters of mercury. And this guy is going to push fluid out of the capsular space into the glomerulus. So let's fill this in on our little picture up here. So if we are talking about something of the capsular space, do I want to pick this one up here that's residing in the glomerulus, or this one down here that's residing in the capsular space? I want to pick this guy in the capsular space. So HPC, that hydrostatic pressure of our capsular space, is going to push fluid out of the capsular space into the glomerulus. All right, our last pressure here is going to be that colloid osmotic pressure of the glomerular capillaries, or the glomerulus. So remember, our osmotic pressures are going to be set up due to the presence of proteins. There's definitely still going to be proteins in the blood of our glomerular capillaries because it's really all that's kind of staying in there. So this is also an inward pressure that inhibits filtration, and this is going to pull fluid back into the glomerulus. So let's fill this guy out on our diagram. It's the last pressure down here. It's inward, so it's going to be blue. So this guy, the colloid osmotic pressure of the glomerular capillaries, is OPG. So this is going to pull fluid from the capsular space into the glomerulus. So I know this is our last box. Hopefully you know where to put it. So OPG, that colloid osmotic pressure of the glomerular capillaries or glomerulus, is going to pull fluid from the capsular space into the glomerulus. So these are all of our pressures. For exam purposes, I want you to know that outward pressures promote filtration, inward pressures inhibit filtration, and then I want you to know specifically what the outward and inward pressures are. Remember, hydrostatic pressures are always going to push fluid out of whatever space that little subscript is, and our colloid osmotic pressures are always going to pull fluid from whatever that pressure is as well. All right, let's talk about net filtration pressure. We will still be using this diagram. There's one thing left. So this is how we are going to calculate net filtration pressure, is based off of those inward and outward pressures. So looking at this here, net filtration pressure equals that hydrostatic pressure in our glomerulus subtracted by the colloid osmotic pressure of the glomerulus and the hydrostatic pressure of the capsular space. So looking at this here, these two pressures are actually our inward pressures, and this guy right here is an outward pressure. So the reason why we don't have the other outward pressure, that colloid osmotic pressure of the capsular space, is because that's relatively zero. So if we plug in all the numbers that we just discussed, that hydrostatic pressure of our glomerulus is about 60 millimeters of mercury. Then the colloid osmotic pressure of the glomerulus is about 32 millimeters of mercury. And the hydrostatic pressure of the capsular space is about 18 millimeters of mercury. So if we do all of this math here, we get positive 10 millimeters of mercury. So if we get a positive number, that means we are having filtration occur. And this is a relatively normal net filtration pressure. So let's go ahead and fill this in real quick down here. So net filtration pressure, or NFP, is going to be equal to our outward pressures. The only one that we're taking into account is the hydrostatic pressure of our glomerular capillaries, subtracted by our inward pressures. And this is going to be the colloid osmotic pressure of the glomerular capillaries plus the hydrostatic pressure of our capsular space. You guys might want to draw this lower down on your diagram. So what this is showing us is that we are having about 60 millimeters of mercury that's pushing fluid out of the glomerulus into the capsular space. And then we are having, let me fill these out individually, you guys don't need to know these numbers, but total we are having about 50 millimeters of mercury that is pushing fluid into the glomerulus and out of the capsular space. So overall, this is going to give us 60 minus 50, which gives us a net filtration pressure of positive, I'm going to write this in orange, 10 millimeters of mercury out. And this is promoting filtration. So on this last little box up here, you can write net filtration pressure equals 10 millimeters of mercury. And that is going to be a promoting filtration pressure. So overall, know basically all of this stuff. You don't need to memorize the numbers, but you do need to know that overall we do have a positive 10 outward pressure that is promoting filtration.